me start with this terminology, translational bioinformatics. Usually this means that research results are translated into practical applications. But we also use this terminology for sort of in the opposite direction of translation. So we target the society and then we help to understand scientific basis of disease and drugs. I can show you with this slide in a better way. So we are interested in this genome sequence data, and we help to extract practical values that can be applied in many things in society. This is a regular meaning of this translational bioinformatics. But we target this society and then provide drug information service. Actually, what we do is that we have a drug label information of drug labels of all the marketed drugs in Japan and also in the USA. So this is a kind of a legally binding documents for actual when people actually use drugs. So by doing this, we have a huge increase. So this is a kind of web access statistics to our website, this KEG website. And since we started, we have a huge access from Japan. These brown ones are from Japan and blue are from abroad. And we have over 1.5 million unique visitors per month to our website. So, many people in society, including pharmacists, doctors, consumers, and patients are now using our resource, and some of them may actually reach more scientifically oriented database of various types of KEG database. So the resource we have is called KEG Medicus. This is part of our KEG resource, and I would like to show you that by doing this type of service, so this is so I think this conference is focused on this translation, but so this is a collection of drugs that are already successfully developed. But we do, in addition to providing individual drug label information, we also perform a comprehensive analysis of all drug labels, especially in Japan, so that we can extract some types of knowledge, especially on drug interactions. I also briefly mentioned about this antimicrobial resistance that's related to pathogen genome and drug response. Before going more detail about drug interactions, I'd like to just give you an overall picture of our entire KEG resource. KEG is an integrated database of 17 major databases, plus these drug labels now. These are actually outside databases, but we have 17 internally developed databases that are categorized into systems information, genomic information, chemical information, and health information. So so these are all color-coded. So systems information, you may know these KEG pathway maps. They are manually curated reference knowledge base. So we have a collection of this reference knowledge base. It's an experimental knowledge on systemic functions of the cell, the organism, and the ecosystem that are represented in terms of molecular networks such as KEG pathway maps. And the main use of this resource is to do the interpretation of genome sequence data and other high throughput data by using this as a reference knowledge base. We also have a chemical database, chemical compounds and reactions, and also this translational bioinformatic resource, including disease and drug information. Now, the main part is this molecular networks in KEGG, and there are three types. KEGG pathway maps represent gene-protein networks. We also have chemical reaction networks that are relevant to metabolic pathways, and we now have these drug interaction networks. Important thing about these KEGG molecular networks is that we collect individual instances based on experimental data, but at the same time we try to generalize these individual instances into classes, so that when you have a new instance, then by checking against these class information, we can make predictions. These classes are called KEGG orthology for gene-protein network, reaction class for chemical reaction network, and we are now developing these drug groups for drug interaction networks. And there are also functional units called KEGG modules, reaction modules, and interaction units for these three types of molecular networks. Let me give you a few examples, but let me start with this the concept of this KEGG orthology. This is a type 2 diabetes mellitus KEGG pathway map. We draw these types of maps based on experimental data, but experiments are of course done on using specific organisms, perhaps mouse, rats, and sometimes human cells. But when we draw these maps, these boxes represent proteins, but we generalize these individual experiment data from specific organisms to ortholog groups. So, for example, this is one ABC transporter that is generalized into this ortholog group K number 05032, and but actually this corresponds to specific genes in specific organisms. So by developing this type of generalized pathway maps, when you have a new genome sequence, then you just compare against this type of generalized maps so that you can generate organism-specific pathway maps that will enable interpretation of your genome sequence data. KEGG module is a conserved unit in the pathway maps, mostly. This is a TCA cycle, and TCA cycle actually consists of two KEGG modules, the first part and the second part. And module is represented in terms of the logical expression of KEGG orthology identifiers. I don't go into the detail, but this segment is far more conserved than the second segment when you check all the completely sequenced genomes. This particular segment, this module is modules of genes and proteins information. So what kind of enzymes, enzyme genes constitute this segment? But you can also see this segment in terms of reactions, how chemicals are converted by specific enzymatic reactions. Then this segment corresponds to what we call reaction modules, this one. And actually we do a kind of reaction data processing, and again, I don't go into the detail, but there is a concept of this reaction class. It's like an autologous reactions. So meaning that reaction patterns are identical, but the actual substrate structures are different. So although the overall structures are different, but the local chemical structure transformation patterns are identical. That's the concept of this reaction class. So by using this concept, so we can generalize very many different types of reactions. We haven't done that in an extensive way, but we are checking the drug metabolism, chemical structure transformation patterns, for example, by CYP and other enzymes, and making that type of information more predictable. So this is just this is just one example of this reaction module, consisting of, in this case, 4 reaction classes, and the top one corresponds to the one you saw in the TCS cycle segment. But actually, there are many different other reaction modules that appear in different pathway maps, but having the identical chemical structure transformation patterns. And this slide just shows you the relationships between gene-based definition of KEP modules and reaction-based definition of reaction modules. The module I was showing you is actually an extension of 2-oxocarboxylic acid chain by tricarboxylic acid pathway. And this portion corresponds to the first half of the TCS cycle, but actually there are other patterns, extension and modification patterns that are related to different, so these are the identical reaction modules, but corresponds to different modules. I was just trying to say that KEP modules and reaction modules are very highly integrated in certain cases. Now, let's move to drug interactions. But this is not a really real drugs, but these are natural products. But of course, as you know, this penicillin and the cephalosporins are originally natural products synthesized by fungi and actinobacteria. In our KEP pathway map drawing, this shows the biosynthetic pathway, and this diagram shows peptide glycan biosynthesis. And as you may know well, these antibiotics, beta-lactams, inhibit penicillin binding proteins in the peptide glycan biosynthesis pathway, so that the cell wall biosynthesis is inhibited. So this is the major molecular mechanism of how beta-lactams work as antibiotics. But of course, the bacteria in this, so this is the bacteria, so this is a different bacteria or a fungi. So this type of interactions are not currently specifically represented in the KEP pathway maps, but we are trying to identify these types of interactions as drug interaction units so that they can be integrated into the existing molecular networks. In this particular example, so this bacteria who are attacked by beta-lactam antibiotics have different ways to evade from that type of attacks. So it's a resistance pathways. Penicillin binding proteins can be mutated so that beta-lactams can bind, or beta-lactamases that are phylogenetically related to penicillin binding proteins can degrade, hydrolyze the beta-lactams, or this is a outer membrane protein for gram-negative bacteria, or this one is exporter. So by decreasing penetration or increasing efflux, the bacteria can escape from the attack of beta-lactams. So by considering these molecular mechanisms, we define drug interaction units. One major thing is that interaction of drugs and targets. The example I was showing you is beta-lactam and penicillin binding protein. But at the same time, the bacteria can hydrolyze or transport. So I use the terminology drug metabolism, but it's a very wide sense how the drugs are processed within the cell. So this one was a bacterial cell, but these two examples are human. Warfarin, anti-blood coagulation drugs, targets this protein in the vitamin K biosynthesis, but at the same time this drug is metabolized by this CYP enzyme, and as we may know, depending on the polymorphism of these genes, some people have poor metabolism, cannot metabolize well this warfarin, so that you have to consider the type of personal genome information when prescribing how much dose this warfarin should be given. Drug metabolizing enzyme can also be the cause of this drug-drug interaction, adverse drug interactions. This particular drug is an CYP3A4 inhibitor, so that when using this is one type of antibiotics, and this one is anti-migraine drug, but you have to be this one is written in the drug labels that these are contraindicated examples. So drug interaction unit is a unit of drug target interaction and drug metabolism in a wide sense. But by defining this type of units, we can integrate the drug information, how drugs interact with our body, cells and organisms. Or in other words, we have an existing wide variety of pathway maps, so this information can be integrated into there. By developing that type of data sets, and also we are developing some tools that might be useful for practical applications. For regular users of our KEGG resource, mostly they have a genome sequence or other high throughput data, and they compare against KEGG pathways and other molecular network database so that they can interpret the high-level functions of their original data sets. We are now developing what we call pathogen checker. So if you have a completely sequenced pathogen genome, then we will check whether this genome contains what types of drug resistance and pathogenicity and so forth. We have a drug interaction checker. So given the list of drugs you are taking, we'll tell you there aren't any adverse drug interactions or not. And for this type of checking, we may eventually include this personal genome information, especially for drug metabolism. I'd like to give you some more details about this personal genome and drug interaction. And at the end, I will also mention about this pathogen checker. Now, in the FDA drug labels, there are some descriptions about gene variations, polymorphism that are relevant to drug response. And we classify this type of polymorphisms or variations into two types. One is this companion diagnostics. Of course, this one is very well known. So many cancer drugs are effective to a certain limited number of patients who have a specific genotypic and other types of patterns diagnosed by specific markers. One example I show you is this crizotinib. It's lung cancer treating drugs that's related to this EML for ALK fusion genes. But this drug is crizotinib and Zalcuri, it's a trade name. And this is just one example of our KEGG drug information. This is we have a collection of all drugs marketed in Japan, USA, and Europe that are combined in terms of the active ingredients so in terms of the chemical structure of active ingredients, and that is then associated with target information and relevant KEGG pathways, drug metabolism, drug interactions such as CYP inhibition and CYP induction, as well as various types of drug classifications. For this particular drug that is linked to this KEGG pathway map, non-small cell lung cancer, and this is the fused gene. So this is the target, first identified by a Japanese person. We also have a somewhat different map called KEGG drug structure map, showing a history of drug development, in this case protein kinase inhibitors for cancer treatment. Crizotinib is, in terms of the molecular mechanisms, this imatinib, it's for leukemia treating drug. This is also a fused gene, targeting fused gene, and it's somewhat related. So this companion diagnostics is the one type of utilizing personal genome information, but the other one is a more subtle polymorphisms and germline mutations, and many are related to drug metabolizing enzymes. I showed you one example, this warfarin. This one is related to both target polymorphism and also metabolizing enzyme polymorphism. And FDA drug label gives a specific recommendation about the initial dose of warfarin depending on the SNP types, polymorphisms of target and metabolizing enzymes. We have, of course, a KEC pathway map, how this target works for vitamin K cycle, and that's then related to this coagulation cascade. We are mostly focusing only on the second type of gene variations, because the first type is of course it's a more well defined, and it's relevant for doctors, but there is not much what we can do. But for the second type, especially related to drug metabolism, we might consider including that type of data sets for analyzing drug interactions. So this one, so this is just a listing of a few examples for polymorphism, but we will be adding this type of data more. There's a tool called Drug Interaction Checker on our website. So if this is only for the US, drugs marketed in the USA, but you give a list of product names, then we'll check if the list is okay in terms of the drug interactions. Whenever we find contraindications and precautions, we report. This particular example is I already showed you, but it's a macro contraindication between macrolide antibiotics and anti-migraine drug caused by this CYP3A4. Because this one is a strong CYP inhibitor, and this may lead this anti-migraine drug not metabolized, leaving the drug at a high blood concentration. In the Japanese version, we have a better interface, but whenever you have interactions, we can also make links to the original drug labels that describe the types of adverse interactions. This is not limited to only Japanese version, but English version as well. But in addition to drug interactions, we also report possible drug overlaps involving either same active ingredients or similar efficacy, different active ingredients, but similar efficacy, such as I think H2 antagonist. These drug interaction checkers are based on the drug interaction database that we are developing. The information, the original data, comes only from the Japanese drug labels. So we do a systematic analysis of Japanese drug labels and extract drug interactions associated with contraindications and precautions, and then standardize and convert drug names and drug group names into KEC identifiers and annotate whenever possible these interactions in terms of CYP enzymes or transporters or targets and other information. This just shows you our internal database for one type of one drug label and how we do the annotation. But just give you an idea of how much data we currently have. We have over 200,000 interactions, but most are actually unclassified in terms of molecular mechanisms. Whenever we know CYP and other types of interactions with this, we add additional information. I'd like to mention a little about this antibiotic resistance. CDC, Center for Disease Control in the United States, give what they call threat levels to these different types of bacterial strains. Urgent threats, serious threats, and concerning threats. We have been developing a resource to enable sort of prediction of bacterial strains that might be classified into these threat levels based on genome sequence data. To do that, what we call signature modules and signature KOs play the major roles for this type of prediction. Signature modules are one type of KEC modules I showed you at the TCA cycle example.
Sorry, the 5 minutes.
So this is for methicillin resistance. So if you find these gene sets in the pathogen genome, then that bacteria must have this methicillin resistance. These are 3 genes, and this colored means that they are adjacent on either the chromosome or plasmid. And we have a list, we did analyze the organisms in KEG, and this is a list of organisms that are classified at this threat level according to CDC. A similar thing can be done for vancomycin resistance. But so these are the cases where mobile elements are involved in spreading this antibiotic resistance. But there is another, and this is all the list of those resistance modules, mostly mobile elements, how they are related to different threat levels. But there is another type of major mechanism for causing antibiotic resistance that is gene mutations. And this is especially true for beta-lactam resistance in gram-negative bacteria. And we collected over 1,300 gene variations that are linked to specific resistance and that are categorized into orthogonal groups, KEGG orthologies. So 1300 classified into 43. And by using this, we made a listing of what type of threat levels these genes are related to and what types of drugs these are related. For example, these are carbapenem resistant genes, and some are, and this is just shows you the phylogenetic tree behind these type of classifications. And to check the validity of this approach, we also checked against the existing genomes for the presence of specific types of mutations. And this is a prediction we make for carbapenem resistance proteobacteria that are classified at the highest urgent level, threat level, according to CDC. So we now have this KEGG pathogen checker. This is not, this is a very preliminary version, but if you give a genome sequence of pathogens, then we report possible antibiotic resistance. So just to summarize, for drug interaction networks, so drug data are used in society, namely drug levels are integrated in KEGG. So this was used to obtain comprehensively the drug interaction data, and some are now linked to polymorphism and that type of personal genome information. For antibiotic resistance, we collect pathogen genomes that are then linked to the antimicrobial resistance. So I'd like to just thank my collaborators. Thank you for your attention.